So I was presenting the work of my lab, and this is a research work on chromosomal abnormality, which is deletion chromosome 6q. So it is known for a very long time. It has been described 40 years ago. But because it's a large chromosomal deletion, the molecular target were not identified to date. And we have been doing a huge genomic work to characterize the minimal region of deletion, and we identified 2 genes that are recurrently deleted in all cases with this translocation. And we understood how it works in terms of mechanistic, because these 2 genes are involved in ribosome, messenger RNA stability, and the fact that they are lost push the cell into a state which is to be more dependent of the glycolysis with respect to the mitochondrial respiration. And the stem cell, are more strong, are more prone to reinitiate the disease with that abnormalities. And we showed that by mice model profiling and by doing xenograft transplantation in mice. Yeah, so the application are potential, and we can imagine to use this pathway, to target this pathway, to modulate the balance between glycolysis and mitochondrial respiration in this cell. But this balance is very subtle, so that won't be very easy. It would be to be just a good targeting of these cells. So that might be not that easy. We have trials in preclinical models ongoing in mice. So we established the leukemia in mice, and the aim is to see whether or not we can treat the mice with that before to going on to patients. Another possibility would be to use this deletion as a vulnerability, because when you delete a chromosome, you lose half the copy of the genes. Some of the genes are not at all relevant for the disease, but because there is only 1 copy, there is less. And then you can target these non-relevant genes in a way to go for a vulnerability of the cell. So this is something we study in the moment. Yeah, so this is the case. We haven't showed that yet. It is not in our paper, but on these 2 genes. But actually, when we look the databases, this gene can be mutated in other cancers. It can be mutated in acute myeloid leukemia in a different way, meaning that these genes are lost in our acute lymphoblastic leukemia, the leukemia we study, but in other leukemia, myeloid leukemia, they can be gained, because it's a kind of balance. So they are pro-oncogenic when they are expressed in some leukemia, and they are pro-oncogenic when they are underexpressed in other leukemia. So we plan to study that. So yes, there were some other presentation in the session, and the aim of this session was to make the link between the fundamental research and the translational and the treatment. So there was a very interesting presentation from Jan Kool's University of Leuven, and he showed that actually he identified with his team a pathway which is downstream the Notch signal in acute lymphoblastic leukemia of the T cell type. And we have inhibitors of Notch that should be very potent, but that failed in clinical trial because they have too much toxicity. And what Jan identified is that one of the downstream protein can be targeted, and this protein is expressed only in the leukemic cell, meaning that when he targets this protein, then only the leukemic cell will be targeted without toxicity. So he showed that in a preclinical model, meaning in animal, and it is really very promising for therapeutics. So all the clinicians that were there, all the physicians heard that, and they are very interested to go into clinical details. The paper will be out in 1 week. Then we heard novel insight from Juan Barata in Lisbon and Peter van Vleerberg in Ghent. And both of them, they studied the transduction signal in the acute lymphoblastic leukemia T-cell type as well, depending of the interleukin-7 receptor. And they show that we can imagine how to target this cell. So it's not yet in clinic, but it should be in a few years, let's say. It's not an easy task to transfer the research into clinic in acute lymphoblastic leukemia. And the panel discussion was really dedicated to that. How can we transfer all the knowledge we have in genomics, and now we really have a very comprehensive view of how it works, how the cell became tumoral and stay tumoral, stay aggressive, but the transfer into clinic is not that easy. So it was a kind of brainstorming how we could do that, and we have to work more. We have to use more a preclinical model. One of the problems is that these are rare patients, so altogether we have to put the patient together, make homogeneous treatment to be able to really evaluate what happened in those patients. And then there was a presentation, not that session, but another, from Jean-Pierre Bourquin in Zurich. And what Jean-Pierre does is for all the resistant patients for the protocol, those with a resistant disease, to put the cell in a media and see whether they grow when they are exposed to many drugs. Huge number of drugs, the drug you couldn't test in all patients, you couldn't test all these drugs in all the patients. So expose the cell like an antibiogram, but a chemiogram, and test which drug would be active on which cells, and try to pick up the good drug for the good disease.